Well, it's 11 o'clock, so we might as well get started. We got people still coming in, but we'll get the introductions out of the way here. I'm Bruce Potter. I'm an integrated pest management specialist with the University of Minnesota Extension. I'd like to welcome you to Southwest Research and Outreach Center Advancing Ag webinar. This is a little bit different format to replace our conventional in-person field days, but what we thought was we'd maybe spend a little more time and go in depth on particular topics. We'll do these hopefully through the season. The first topic we selected was soybean pest management, pre-plant decisions, and what we're looking at is how to balance host plant resistance, chemical decisions, risk, that sort of thing. We want this to be interactive. We've got some good panelists here to help answer questions. We've got a wealth of research in particular on this subject, and maybe some of the questions could help focus some new research. So if we start out with panelists, we've got Aaron Lorenz. He's associate professor in soybean breeding in the Department of Agronomy. Bob Koch. Aaron, I'll give you each a chance later on to make a brief introductory statement. Bob Koch is an associate professor and extension entomologist in the Department of Entomology. Jim Kurle is an associate professor in soybean pathology. Dean Malvick is also in the plant pathology department. He's a professor and extension plant pathologist. And last but not least, we've got the Swiss Army knife, Seth Naeve. He's an associate professor and extension agronomist. So agronomists are supposedly multidisciplinary, so that's why I'm calling them a Swiss Army knife.
Overrated tool, right?
Yeah, they usually break too. All right. Somebody already asked a question on where the coffee and donuts. They're there. They're virtual, so they're pretty inexpensive. So you can take 2 or 3 if you want. Emily, can you put that next slide up? All right. This kind of spaghetti-looking diagram is kind of how people might need to look at some of the factors that need to be considered when they're deciding on soybean insect and disease management before planting. Okay. Risk is probably one of the big drivers. Everybody's a little bit different. There's some ways you can determine risk. We'll hopefully get into things like field history, variety decisions, management options. Some of these pests we might touch on today don't really have any good management options. If they do, we've got things like variety selection, seed treatment, pesticides, things we can do with planting dates and planting and seeding rates. Some of these decisions have to be made up front, things like seed treatments and variety selection. Some can be deferred to foliar treatments and scout and spray later on. And then the other thing that makes this a little bit more complicated is as a grower or a person advising a grower, we've got multiple pests in a field at one time. So this may look a little confusing, but it's things that people really are taking into consideration before they plant. Sometimes they don't, and then we have some problems later on in the season. So let's get started. I'm going to go through the panelists here. First one, we've got Jim Kurle. And Jim, do you want to just make a brief statement on what you think, a sentence or two on what you think is really important in planning for pest management early in the season?
Well, I think we really need to consider the environmental situation, the weather, especially, at least for the pathogens that I've been interested in. They're very much influenced by high moisture situations. And that includes all the oomycetes, Pythium or Phytophthora or Phytopythium, and SDS as a disease. It's also very much influenced by wet conditions. But there are management options too. There are some good chemical controls available now. For the oomycetes, there are multiple chemicals that are available. As far as the oomycetes, particularly Phytophthora, it's probably important to consider maybe combining management practices. So looking at tolerance to Phytophthora, for instance, but also using a seed treatment. There are a number of advantages to that. One of them is that there's some indication that if you are using tolerant varieties, you may be increasing the number of pathotypes that are developing. But if there are seed treatments available to suppress those populations, you can slow that development of pathotypes. And then the seed treatment for SDS Illevo has proven very effective, and there's some recent economic analysis that indicates that even in very low incidence of SDS, it's still worthwhile as an application. And it's probably useful to put in combination with partial resistance to SDS. So I guess I'm turning into an advocate for seed treatments in most situations for these 4 pathogens that I just mentioned, the oomycetes and SDS.
Dean, we might as well stay on the disease side for a little bit. Do you have a sentence or two you just to set the conversation up?
Yeah, I mean, Jim made a number of very good points there. The other thing I would like to add is, I think, the more we can know about the risks in an individual field, the better off we are. If there's no expected risk of SDS, we may not want to use a seed treatment, for example, and that applies to others as well, because some of them are quite expensive. But this applies to all the different pests and diseases, right? If we know where the higher risk areas are, we can more optimize where we use the seed treatments or other practices or resistant varieties, if that's our choice. So sometimes we can know what the risk is for particular diseases, and sometimes we can't. But when we can know, that can help make those decisions. And I'll stop there.
Yeah, if I could just add to that, I agree with what Dean has said. It's another component of it is really understanding what the history of the risk history of a field is. And then in terms of seed treatments like Illevo, which is fairly expensive, it might be worthwhile to develop some techniques for mapping the occurrence of the disease in the field. That's something for the future, but it's something worth considering. Mm-hmm. It would be a really worthwhile application of precision ag techniques if we could map the occurrence of SDS in a field, for instance. So we would really be economizing on the use of the seed treatment. But Dean made a good point. It's important to know what the history of the field is and what probable disease occurrence would be on that field.
Bob, do you want to address some pre-plant decisions for insects real briefly? Try to keep these short because we're getting quite a few. We're getting some questions coming in, so this should be pretty lively.
Sure. So I think from the pre-plant end of things, we're really kind of limited in what we're doing for soybean insects. We've got seed treatments like Jim and Dean were talking about. We've got seed treatments for insects. And I think we need to think about those field histories and specific field conditions where those are most likely to pay off. As for, varietal selection, there are effective aphid resistant soybean varieties, but they're not very widely available. So I think because of these 2 factors, we're really relying on the later season foliar applications of insecticides, and we can chat more about some of that later.
Aaron, you want to talk a little bit about breeding and some of the complexities on deciding what we need to be worried about?
Sure. I agree with everything that's been said, especially about knowing your field history. And there's a lot of variation out there among commercially available varieties for their levels of resistance and tolerance to these certain pathogens. And then I'd say the availability and usefulness And I guess reliability also of the ratings given out commercially depends on a few things. It depends on how prevalent those pests are and how important they are for producers. It would depend on the availability we have for certain bioassays we can use to screen for tolerance or resistance to these pests. Some things are easier to screen for than others. And it also depends upon the genetic complexity of the of tolerance or resistance to the certain pest. So, there's things that range from SCN resistance, which is relatively genetically simple trait and pretty easy to screen for using DNA markers and greenhouse bioassays, to things that are much more complex, such as SDS resistance and white mold resistance. So these ratings are available in seed catalogs from commercial varieties, from commercial companies, and getting a handle on that for your specific situation is certainly an option. So.
Okay, Seth, our Swiss Army knife, what do you think about the situation?
Well, I mean, obviously, I go back. I think a lot of this we could actually focus back on that figure of yours, and I think as I look at it, I think the idea would be to think about the weighting of those various lines and where the most impact comes in. And I think the biggest issue that I see with farmers and decision making is weighing that risk. I think there's a disproportional view of risk versus the efficacy of some of these products, and farmers are for very, very good reason, are very concerned about reducing their risk. Unfortunately, I don't think we've done a good job helping them measure what their actual risk is relative to these products that we're putting down. And so, I think it's a global thing. It's, on a whole national scale, it's field to field variation. And then Jim mentioned it's within fields. So, we get to the point where at some point, if we know that there's 2 or 3 acres in a specific area that need treatment, then we can make really educated decisions about how much to put on the whole field as a blanket versus how much to put in a certain area and how much effort. And at this point, we're a long ways away from there. We're still treating 60 million acres of soybeans with fungicide, insecticide seed treatments, 50 million acres maybe in the US, and maybe 2 million of them really require that. So I think we're missing the boat a little bit on risk and how we respond to that risk. And so that's the biggest challenge for us at the university, is how to deal and how to communicate risk to farmers. So I think that's part of the future, is better understanding that piece of it. So I'll stand down.
Okay, thanks, Seth. Before we start answering questions and get further into the webinar here, we've got to do some housekeeping. And like I mentioned, this is the first in a Southwest Research and Outreach Center's Advancing Ag series. And we're intending this educational series to include monthly events that we want to bring together a diverse panel of researchers and specialists, and we want to address crop production topics from multiple disciplines. We've pre-submitted questions already, and we'll be addressing those, but we'll also take questions live. So please submit your questions through the Q&A feature that's on the bottom of your screen, and type them in. We may ask you to unmute at some point, but we're going to try to handle most of these through question and answer. So we might as well get started with questions here. First question we've got, I've got here deals with soybean cyst nematode. And the question is, how do we reduce SCN more effectively? In other words, in order of impact, what strategies best combat SCN? Personally, I think the big bullet right now is still crop rotation and resistant varieties. But anybody want to tackle that question in the panelists?
Well, I think that one component of that would be making sure that you know the actual SCN resistance of your variety. I mean, there may be something that's labeled as being resistant, but there may be some actual reproduction on that variety. So you can look at state variety results and things like that. If that variety has been entered into those SCN bioassays, you'll actually get a female index that relates to how resistant the variety actually was reported for that variety. So that's one thing to look at. And if you have a favorite variety out there that is supposedly SCN resistant, you want to actually quantify that level of resistance, you can feel free to submit it to us and we'll get it bioassayed for you. So making sure there's actual resistance there is one part of that. And then over the longer term, I'm sure people have heard a lot about this issue of resistance breakdown on the 88788 sources of resistance. To kind of combat that over the long term, you can rotate away from an 88788 resistant variety or maybe soybeans altogether for a while, and then rotate in your sources of resistance, whether it be an 88788 and a Peking or something like that, to try to keep those populations of SCN more stable.
Jim or Dean, do you have any thoughts on some of the nematicide seed treatments out there? How effective rotation is? And Aaron, any thoughts on how far away we are from new sources of resistance? Maybe start with that to follow up on your comments, Aaron. How easy is it to breed that resistance, and how far away are we from getting new stuff into the system as far as managing SCN with resistant varieties?
Well, there are a number of commercially available Peking types already out there. And I think, the number is still small, but I think the number is slowly ticking up because I think that in some of these places where we do have resistance breakdown, the Peking types are really shining through. And so farmers are really starting to ask for those in some regions because they're seeing the yield advantage. So that's a positive turn of events. And then there are some other additional sources already out there on the marketplace. I think Golden Harvest right now has a novel source of SCN resistance that's commercially available. We had that in our variety trials last year, and it did pretty well. So that's out there. And then in terms of the sort of more in the discovery phase, we have lots of things in my own program that are in the pipe that have different sources of resistance that are maybe in their third or fourth year of yield trials. And but in terms of overall how long it takes quite a long time between identifying a new source and breeding that into a commercially acceptable variety. So it's a long-term process. We're talking about a decade or more. So we've been working on it for a long time, though, fortunately.
So in other words, I'll be retired before anything exciting happens. Is that what you're saying, Aaron?
No, I think there are exciting things happening right now. It's just that they were initiated a number of years ago. They weren't initiated last year or anything.
All right. Dean and Jim, you both talked about seed treatments, and we've got a question here. Does the wide use of seed treatments help build up resistance? In other words, we'll start with SCN, but this actually goes through other soybean pathogens as well, not just SCN.
I can start with that, Jim. Just a few comments. In my opinion, now the seed treatments used for SCN are, the jury's still out on how well they really work. There's some evidence, some places that there's some activity there, but certainly they're not the silver bullet we were all hoping for a management strategy for SCN. But they might be part of the picture. The bigger picture about do other seed treatments generate resistance or induce resistance to those compounds? That there's some evidence that can be happening. And Jim has some comments on this, I believe he'll have. But overall, the risk of resistance development is certainly lower there than spraying a fungicide widely across acres for a foliar pathogen. But nonetheless, the risk is there, especially as we use these more and more widely, in every acre on every year. I'll stop there.
Thanks, Dean.
Yeah, I'll just follow up on that. Probably the question here is more variety in the treatments we're using. And so metalaxyl mefenoxam have a history of causing the development of resistance in foliar applications and vegetable crops, but there's not real evidence for that as a seed treatment on soybeans. And we're probably developing some insurance there too. There are a couple other seed treatment fungicides that are very effective against the oomycetes. And it would be a good practice to kind of rotate the seed treatments. I'm trying to think of the name of that. Okay, here we go. Ethoboxam is one of them. Oxypipro, I'll take it from there. Anyway, and the three of them are very effective. And when they're used in rotation, I think the likelihood of resistance development by the oomycetes is very unlikely. I don't think we have much information about seed treatments for SDS, but just the characteristic of that disease suggests that resistance is probably not going to be a problem there. Okay. And I'll just make that a very tentative comment.
Okay.
Bruce, can I jump in? Can I jump
Sure.
in real quick? I think
Yeah.
just to broaden it out, I think the issue really is around success of these products, and anything that's successful, we have a history of putting it out everywhere for everything. So I think that goes for, you know, the broad conversation here is that any of these really good products just get deployed everywhere. And, you know, that raises multiple problems. And I guess, you know, one maybe more narrow example of this is some of the foliar fungicides, and then they work so well, let's just throw them on the seed too, kind of thing. So you've got multiple, you've got products that work well. And so then we're going to find new avenues, and those companies just find new products to put those same actives into. And so they can, you know, generate more profit. And, you know, of course we always wonder if it's part of planned obsolescence here, if they're trying to rip through some of these so that they can provide a new product. And I don't know if there's any evidence in that, but it's certainly, if you look at marketing and how other products are made for other industries, it certainly mimics that kind of behavior.
Bob, what about the neonic seed treatments for soybean aphids and other early season pests? We've got a couple questions on neonic registrations, and will we have enough insecticide modes of action down the road? And then we've got the issue of the neonic seed treatments, and then some of the foliar applications. Any comments on that?
Yeah, I think from the regulatory perspective, I'll touch on it a little bit, but I think we've got some colleagues from MDA on the in the audience here. So if I misstate anything, they can certainly weigh in, and I'm not going to go too deep into it, but the neonics are labeled and being used seed applied and for foliar applications, and we still have access to these tools for those purposes. What's going on now with the neonicotinoids is the state does monitoring of surface waters, and in recent years they've been detecting some of the neonicotinoids above certain thresholds, and because of that, it's been declared a surface water pesticide of concern by the Commissioner of Agriculture, and then that triggers some development of best management practices, and the hopes there are to reduce these detections in the surface waters, the frequency of detections and magnitude of detections. And if that doesn't happen, I think the state can increase regulations or restrict use of these products, but we're not there yet. The two neonics that were listed in this declaration as surface water contaminants were clothianidin and imidacloprid. And remember that clothianidin is a breakdown product of thiamethoxam. Most of these detections, I think, were in the surface waters are showing up in June and associated with storm flow, so runoff. So that's kind of what triggered all of that. And you may have seen some announcements coming out from the Department of Agriculture there. In regards to, you know, efficacy, there's a lot of data showing that the neonic seed treatments can be very effective at protecting soybean seed and seedlings from pests when those pests are there, right? So kind of reiterating what Seth was saying, these soybean seed treatments are being used very widely and we may not be getting benefit from them on, you know, a fair size fraction of that acreage, but there are probably certain fields or certain situations that have, you know, conditions that are conducive for these pest populations, and we're likely to get a benefit from these seed treatments. It could be seed corn maggot, or maybe something early season like bean leaf beetle. As for soybean aphid, the multi-state research that Bruce and I and colleagues have been involved with, you know, Seth, I think, has been involved with some too. For soybean aphid, it's still suggesting that scouting and use of the threshold for foliar applications is probably a better approach for soybean aphid management. However, if you've got a situation where soybeans are being planted late, maybe after harvest of peas, it might make sense to use seed treatments there, because you've got higher risk for soybean aphid populations showing up there while the concentrations of the insecticide are going to be still high and effective in the plants. And they might also be useful in some fields where they get colonized historically earlier than others, you know, and maybe that's smaller fields in river valleys near a lot of wooded edges with buckthorn. And again, the idea there is early in the season like that, if they're getting colonized, the concentration of the neonics are probably still high enough in the plants to be effective. But under a typical colonization pattern, the insecticide kind of peters out before the aphids usually get there.
Okay, what about modes of action, Seth? I got somebody just texted me, said I look pretty bored, but that's my paying attention face. You just don't see it very often. Are we going to be all right on that, or we got new? You know, we've got diamides coming out and some other things. So, what's your feel on just overall? If a guy decides not to use a seed treatment, are you going to be okay on the back end for things like soybean aphid or bean leaf beetle or any other soybean pest for that matter?
Yeah, I'm not sure I quite follow your question, but I think, are you talking about just a pure prophylactic application of these things? Is that your question?
No, I'm talking about do we have enough different types of chemistries available for insects and soybeans, different modes of action, that if a person decides he's not going to use a seed treatment, that he's got enough good options later on for things like soybean aphid with resistance and other insect pests as well. We think of bean leaf beetle as an early season pest, but is it possible to control that without a seed treatment?
Well, I think Bob's the right guy to take this on, but I would say that there certainly is. And again, I keep going back to this broad issue, is that repeatedly through a lot of our research and looking at these, at the work, when we look at pure prophylactic applications of products without any knowledge or any idea of potential increased risk, we don't see value in these things. It just, or if there is, it's very, very minimal. It's only enough to pay for the, to break even on the ROI. So it goes back to this point, is that we just have to know, we have to have some information about risk in these fields in order for us to move that direction. And in a really broad sense, we have to have ideas that there's increased risk in order for us to put out the vast majority of these products. And I think that kind of leads into your question, is that, should we rely on scouting and taking care of these things after? And I would say that for the most part we can, but I think Bob needs to confirm that for me.
Yeah, for soybean aphid, like I said, I think scouting and foliar applications based on the threshold is the way to go there. As for the number of insecticide groups or modes of action available for soybean aphid, which is the pest that's probably getting the most foliar applications, we've got 4 groups there. Group 1, which is the carbamates and organophosphates. Group 3 is going to be the pyrethroids, and we've got the resistance concerns there in soybean aphid. Group 4 has the neonics, the products like sulfoxaflor and fluopyridiferone. So some of the newer products, Transform, Savanto. We've got potential regulatory issues around the neonics. The other 2 are effective. And then we've got the newer group 9 product, iphidopiropin. So we've got 4 insecticide groups available for soybean aphid. We've got some resistance concerns and regulatory concerns that are kind of whittling that list down. There are other effective insecticide groups against aphids, but they're not labeled right now. But it's something that maybe could be considered in the future if this list gets reduced anymore.
This is kind of a pretty holistic question here, and kind of the point of why we're trying to do this format. We've got a question in here. What does research tell us about the value for pest management of extended rotation intervals? So, and I think you all have some input on that as far as how well can we combat insects and diseases, things like white mold or SCN, which persist a little bit longer, some things that are shorter duration. How well are we using rotation to combat some of these problems?
Well, we're not. But honestly,
could we?
For sure, we could. And I think, again, this is a question about balance and risk and value of these things. And I'm inferring from the question that the idea is that extended rotations is a silver bullet, and I don't think there are silver bullets. I think it certainly is it reduces risks broadly across a lot of problems, but it doesn't answer any of the problems that we're talking about. And so what we need to discuss is how much it reduces that risk versus other things that we're doing. So I think those are the real questions for us.
So let me ask a question this way. What about things that are going to be problems from year to year, things like SCN, things like white mold every time you have soybeans in that field, versus some of these pathogens that are windborne to a certain extent and can move in from other fields? So anybody have some thoughts on that? Just how do you you said there's no silver bullets. Could you maybe phrase that in terms of what type of pest or disease you have?
I'd like to pass that off on someone else, but I would say we have to look at them all individually, and there's biology going on, all of them, and so we have to look at them individually. I think and that's part of the question about rotations, is it does help a lot of things, but it doesn't solve all the problems equally, is another way to look at it. So maybe Jim and Dean have a better answer for
I would agree that particularly with the pathogens, we have to really recognize what the most important pathogen is. And for instance, white mold, rotation is not the very effective way for dealing with it. That disease is essentially driven by environment, and once it's established in a field, it is very difficult to manage it if you have an environmental situation that keeps contributing to it. You're probably in a similar, it's interesting, there's some recent research, Dean's lab had done some of this. We may have to look in more detail at what we're rotating with when we're considering SDS, because the SDS pathogen is very effective at reproducing on non-soybeans. It increases inoculum in the saprophytic phase. And there's research here in Minnesota and Iowa State that indicates that if there's adequate carbohydrate source, it reproduces very well on corn. So a corn-soybean rotation really wouldn't be an effective way of dealing with that pathogen once it was established in a field. And when you're talking about Phytophthora, there's a very persistent reproductive structure there. So once it's established in a field, if you have the right environmental situation, you could very easily have serious outbreak if you have susceptible varieties. So it can require some really careful consideration of what you're going to rotate with. And if it's practical to even try to rotate to manage the disease without looking at some of the other disease management practices.
Dean or Aaron, do you have any comments on that at all?
I'll just add a couple comments. I mean, that I think all the key points were just hit. You know, every pathogen has a different response to these different rotational schemes. You know, again, coming back to SDS, as Jim said, you know, not only in Minnesota, but various other places, rotation of soybean with corn doesn't suppress SDS. In fact, sometimes they've even shown increased SDS following corn. But more recently, some work out of Iowa State shows some rotation with some legume crops, forage crops, might have some suppressive potential. And that's relatively new and not completely proven as much as it could be, but that's an interesting idea. So that clearly doesn't apply to a lot of situations, but there's a lot we don't know. And you know, the microbes in the soil certainly have a role in this beyond the pathogen. And so if we get to the point where we can start to understand the whole microbiome, the whole complex of microbes in the soil and how they play part in this, you know, we might be able to more tailor this, but we're a long way from that.
Just real quickly from the insect end of things, you know, in corn, rotation is really important for the different rootworms, right? But in soybean, it's not going to be a very effective tool for most of the pests that we're dealing with.
There's another it's not actually a chemical control question, but and it goes back to basically to the seed treatments for the most part. But somebody asked, are there any biologicals that can be used to offset issues of disease or SCM? And that's kind of the I suppose the most rapidly expanding part of soybean seed treatment is some of these biologicals. Anybody have any thoughts on that?
I can make a few comments. I think, you know, a number of the seed treatments for nematodes are biological, they're bacteria. You know, that doesn't say they're all terribly effective, but certainly some of them have some efficacy in their bacteria. They're not a nematicide, fungicide, nematicide or a fungicide. They're not a chemical. So they're out there. In terms of the disease control world, I mean, there's a lot of things being worked on, and only a few of them are available on the market right now. One that's being widely touted and sold, you may have heard about, is called Heads Up, which is actually a plant extract out of a company in Canada, and that's applied to the seed. And that, you know, that has being sold for both SDS and white mold, and I think the jury's out how well that can work for as well. But that's another biological product. But as I said, there many companies are working very diligently on these products, and I've tested some in some experimental field trials, and some of them have looked good, and some of them I did this years ago, and they still haven't come out as products. So I don't know where they are in the whole chain of introduction and marketing. But it's a very active area of research and development for sure.
Can I just jump in and give my own pessimistic view of the biological scene? I think there's tons and tons of money in this area, and this is where all the big companies are spending their money. But from a biological standpoint, from the big biological standpoint, I'm just a little bit pessimistic about a lot of these things, because we're trying, these companies are selling them to put on 80 million acres of soybeans in the US, right? So the problem is the biology of those soils and the physical and chemical composition and moisture content of those soils is very, very different. And so it's hard for me to imagine that these active biologicals are going to have similar, you know, functionality across these environments. And for the most part, they're not building these things to colonize roots. They're producing these biologicals that just deliver these active compounds, whether they're compounds or bacteria, as Dean mentioned, right on the seed. And, you know, the seed placement isn't always the best placement for these things. And so we have a lot of challenges ahead. I think it's a great area. I think there's a lot to, I mean, clearly this is the growth area and there's a lot of potential. But again, back to my silver bullet comment, that it's hard to believe that any one of these things is going to solve problems for farmers universally. I mean, even one problem universally for farmers.
Well, following up on that, and this again, there's some people trying to that are actually putting some different pieces together here. But the question is anyone studying the introduction of foreign microbes and the effect on natural microbes? So the biologicals that we're just discussing, has anybody looked at how those interact with what we've got in those soils, our soils natively?
Well, let me go back and use a real world example as the Pasturia in Clariva for nematodes. I mean, that was pulled out of nematodes as a parasite of the nematode that was found pretty broadly in nematodes, I believe. So Jim and Dean, please correct me, but I believe that Pasturia has been collected in a lot of different locations. And so it's just reintroducing an existing pathogen and making sure that you inoculate those areas with a known pathogen for SCN. So those types of things I think are pretty safe and pretty straight, but this idea of something a little bit more foreign, I don't know that anybody's looked at it much, because I don't know that my concern is that I'm not sure that these things really are very long lived in the soil, but maybe Dean and Jim can add on.
Just a couple of comments there. Another example is Contans, the fungicidal biological control product used for white mold. That's a fungus, Coniatherium minitans. That fungus in Contans is a widespread fungus in soil. I think the strain that's used there is, particularly maybe more active and a bit longer lived maybe than some of the others that are naturally residing in most soils. But again, it's not a brand new organism by any means in very many areas at all. But in terms of the bigger question, are people looking at how introduction of microbes changes the microbial profile of a soil, say around a root system or something? Yes, those kind of studies are going on. In fact, I was at a meeting and there was a public presentation about 4 years ago given by some people from Monsanto, when it was still called Monsanto, and they were looking at very in-depth microbial profiling of soils around plants where they introduced certain potential microbes for biocontrol. And there weren't a lot of results given, but there was a tremendous effort being put in there, and I don't know what's come out of it since, but those kind of work is going on both in companies and in universities.
Anybody else want to comment? So are you saying there could be a zombie apocalypse or not? No, okay. So we've got another question kind of on the same similar topic. We're going back to the treatment of soybeans, but we mentioned the over-treating of soybeans. How do farmers go about making the decision to place the treatment, but also go about treating only partial fields? And the advantage in the insect world is in most cases there's a few exceptions, things like seed corn maggot. A lot of these we can scout and treat where problems occur, but I think it's a good question overall. Where can you treat partial fields, and maybe things like should you or should you not treat whole fields for insects and spider mites? We mentioned treating maybe using a levo on part of the field, that sort of thing. How does a farmer go about making those decisions? Anybody?
Well, we were doing some work that was really evaluating foliar symptoms and using that as mapping an area. We weren't able to take that very far, but that would be one approach for SDS, for instance. And then I think, Dean, you were doing some work on inoculum in the soil. That might be another element that you could add to that symptomology and use it to kind of develop field maps indicating where you're at most risk for developing SDS, or where the inoculum is highest, or where the disease has been most severe. But we're really just at the beginning of that kind of approach. But I think that would be one way to do it, and it would make use of precision ag techniques that are, they're pretty much available. We can map fields. We need to be able to map the occurrence of the disease is really the important part.
Bob, what about the insect side of things?
Yeah, I think, one pest where this, really could potentially be feasible would be for soybean gall midge that Bruce has been doing a lot of work with in the southwestern part of the state. That insect shows a real strong spatial pattern where they're aggregating on the edges of the fields. And, if or when we find effective insecticides for that pest, that could be a situation where we could reduce the overall inputs or application costs by focusing just on those edges. But I think we're a ways away yet. A lot of more research is needed on evaluation of products and especially, application methods. For spider mites, like we dealt with in some parts of Minnesota this summer, a lot of times we'll get heavy infestations on the edges of fields. I think in some situations, edge applications to just the edges of those fields might pay off, but I think it really depends how widespread the mites are in that field. You might be seeing mites and heavy symptoms on the edge, but those mites might have already spread throughout the field and you're just not seeing symptoms yet in that interior of the field. So I think real thorough scouting would be required to make such a decision there if you're going to try to treat the edges of the fields. Historically, I think our recommendations from Extension have been whole field treatments for spider mites. A lot of the other insect pests don't show that level of aggregation, so it might not be as feasible. I know in China, some researchers there that are working with soybean aphid are evaluating the idea of kind of spraying out hotspots that develop in fields early in the season before the aphids kind of redistribute throughout the field. But we'll see how things work out there, and maybe eventually we can do some research here to try to validate that if it shows promise.
Somebody just put a question up. Wouldn't you need a multi-hybrid planter or something like that to be able to plant treated and non-treated seed? Yeah, you'd have to do either plant the field in different chunks or you have to have some sort of technology on the planter, and that's kind of outside my bailiwick for sure. But it's one of those things that might sound simpler than it actually is in practice. Any thoughts on that?
Well, let me broaden out the question a little bit, and I would say that we can. It's pretty clear to me that the folks building the physical machinery and the technology to do those kind of things are way ahead of where we are on the agronomic and the management side. I mean, we can do a lot more things than we should be doing, honestly. So precision ag is way ahead of where we are from a management standpoint. So going back to the original question about how to manage these things by within a precision basis, we have to really nail down the our understanding of the pest and the plant's response to it in these fields, and be able to predict it very well before we're to that point. And so it's a stepwise approach, and I think we're getting closer on all these things. But my feeling is the technology always leads us. We can do a lot more than we probably should be doing.
There's Bob mentioned soybean gall midge, and there's a question here. Where is it at right now? Is it moving northwards in Minnesota? And it's from survey efforts and reports people have submitted from the field, as far north as we've got it in Minnesota, that we've confirmed it as Traverse County. It might be other places at low levels, and for most of Minnesota right now, it's at fairly low. It's present, but at low levels. We're not sure why. We're looking at things like weather. Is there some sort of effect of area-wide tillage practices, something like that? but fortunately at this point, the number of fields with real serious economic problems are limited. That might change. And so we're kind of keeping an eye on it. It's, it's important that if you see that pest in a new area, we find out about it. So we're going to keep looking, and we're going to look at some other crops that might be potential hosts as well during the following year. White mold. Any research on manganese application or soil test manganese levels to assist in soybean white mold control? Anybody, Jim or Dean, have you looked at that at all?
No.
Anybody?
No, we haven't. I haven't anyway. There was some work a number of years ago that was looking at calcium, and there seemed to be some benefit from calcium applications, but I've never seen any follow-up on that.
Dean, have you heard anything about that at all?
No, I've heard people talk about it and ask about it, but I've not seen any data or any results from trials on that.
Okay.
Are we still blaming Roundup for everything? Is that where this comes from?
Probably.
Okay, just checking.
That's right. That's right. There is that manganese interaction with glyphosate.
We'll follow up with Don Huber.
Yeah. All right. Aaron, I've got a question for you. Is in your breeding programs, how do you prioritize what genes or traits you're trying to get into soybeans? How does that decision happen? And then how do you make those decisions by ease of getting it done, or ease or importance in soybean production, or both?
Importance in soybean production, of course, is the primary factor in determining that. So how wide scale the pest is, how frequently it could impact the producer, the risk level it could pose to a producer is clearly the first decision point. And then the next decision point is going to be the source of resistance or tolerance that we can find. So is it something that's easily available in already a commercially viable or elite parent that we can cross into the program and get into our own germplasm? Or do we have to go back to an old land race or something in the collection that's going to lodge and have all kinds of problems? So that's another point. And then also the genetic complexity of the traits and whether or not we can use molecular markers to move in a single gene or two versus a really highly genetically complex trait that we can't do that with, and will take years of breeding to incorporate into a new variety. So those are all decision points for that. And then of course, what other tools are out there to manage that disease? Are there insecticides or other sources of treatment that can be used to manage that disease? Or is the varietal, excuse me, the varietal resistance the only source that, or the only treatment option that we have would be the other determining factor. So those are some things that come to mind.
Somebody just commented that the manganese white mold stuff is coming out of Hefty. So I don't know if that, I'm not sure where their data is coming from, but sometimes you don't need any. Let's see, let's go back to insects for a little bit. And the question, how would you rank 2020 in terms of aphid pressure? And what are the current foliar insecticide application thresholds? And I'm assuming that's for soybean aphids. Bob?
Yeah, I can make a few comments about statewide pressure, but Bruce, Seth, anyone else who had plots out, maybe even Aaron, with the crazy research restrictions, travel restrictions, I wasn't getting around as much this year as before. But overall, I think soybean aphid infestations were lower across the state than in some previous years. And in some parts where aphids did develop, it was later in the season. And I think in some of my research, in the southeastern part of the state over the last several years, it seems like more and more of the infestations are developing kind of later and later in the year, which poses some challenges, right? With pushing up on, when we consider the 250 aphid per plant threshold to be valid, through R5 and some things like that. Seth or Bruce, what were you guys seeing in some of your plots for aphids? Did I capture it okay?
Yeah, I think so. I mean, we had a lot of guys not had to spray aphids in Southwest Minnesota for several years. This year we had the research station center here. We had populations that were at threshold levels, but they kind of stalled out. I think part of it was maybe some interactions with weather and spider mites on some of those plants. But I think the biggest question is getting to be with soybean aphids is what to do about these populations late in the season. And that's one of the reasons why that 250 is there. We want to be sure that those populations have a chance of going up, and that's still a pretty conservative number, I think.
I agree with that totally. This has been my pet forever as a soybean physiologist is that late season, especially dealing with all the seed quality things and the fact that so much happens in soybean in those last, few weeks of seed fill. But I'd flip the question back to Bob and Bruce with this aphids. I mean, what's the 5 and 10 year prognosis for this thing? Is it going to disappear? Is it or just going to continue to wane? And if it is kind of on the downcline over the US, what is it, parasitoids, or what's going on out there? Or am I just mistaken?
That's an interesting comment, Seth. When we think about soybean aphid across the entire Midwest, in the early days of dealing with this past early 2000s, it was truly a regional problem, from Minnesota, the Dakotas, all the way, to the east, out to Ohio. But there's been some research published, and, in talking with my colleagues in those states, infestations have really decreased, to a point, with some of our multi-state research where it's hard for those colleagues in those other states to even come up with an aphid infestation for us anymore to do research. However, in Minnesota, for whatever reason, we're still seeing, fairly consistent infestations. It's going to vary by year which part of the state we're seeing infestations in, but pretty much every year we're still getting a decent chunk of the state where we've got infestations requiring treatment. What's causing that? I'm not exactly sure, though. The one paper that was published was pointing toward, or what's causing the decrease in some of these areas. The one paper that was published is pointing toward the neonic seed treatments and that early season exposure of the aphids kind of knocking down those populations and slowing them down enough. I'm not sure if I totally buy that, because we're using the seed treatments at probably the same rate or same number of acres here, percentage of acres, and we're still seeing soybean aphids. There is a parasitic wasp, Aphelinus certus, that's spread throughout the region and is definitely having some impact on the aphid populations. The level of that impact kind of varies, different research is showing different levels of impact, but we can fairly easily find those black parasitized aphids, the aphid mummies, on plants. So I think it's having an impact along with all the other natural enemies out there, like the lady beetles and other critters.
I think from my perspective, I think actually the intensity of these problems seem to be maybe going down a little bit over time, and one of the reasons is we just don't seem to have that big of area to draw new aphids in to infest. If that'll keep going and continue, or if populations rebound and we end up with more problems again. I'm not psychic enough to know that. So I think the only way we're going to know that is spend some time out in soybean fields. So we're at 12 o'clock here, and I'll stay on a little bit longer if I need to, but I really want to thank the panelists and the participants in this for joining us today. It's a different format, and hopefully it's working for you. But share your feedback. Emily put a link in the chat box. We'd like the evaluation. See if we should continue doing it, or you know, if it's a really bad idea, we'll never talk of it again. And then the next Southwest Research and Outreach Advancing Ag webinar is going to be on February 24th. Jeff Strock is going to lead a discussion on building resilience against extreme weather events. And they're going to discuss soil health and water management. And we'll have future webinars available at z.umn.edu/advancing-ag. So thank you very much.